Okay, thanks for a great talk, Will. Talk was also a great segue into the next talk by Kay Fuller. So Kay is the president of MDRS, the Medical Device Regulatory Solutions. It's a regulatory consulting firm here in Ann Arbor. And Kay brings about 40 years of experience in developing and commercializing medical devices to our consortium. She has worked at Gore, worked at Bard and Terumo, amongst other medical device companies, and she's led global clinical studies for about 40 some odd class 2 and class 3 devices and combination products. She also, there's a question about the CTSA and MyApp. Prior to branching off and starting our own company, Kay helped establish the MyApp, the IND, IDE faculty investigator assistance program at Michigan when the CTSA was launched here. So she brings a wealth of experience and is part of our regulatory team in addition to people in our CTSA. So I'm happy to have Kay speak to us now in the regulatory practicum.
Thank you. It's a real honor to be here with everyone today on this especially cold day for warm hearts. Thank you all for being here. It's a joy to follow Will's presentation. He truly is one of the best in his field in the clinical research space, and it's been a pleasure to have the opportunity to work with him closely in some of his clinical studies over the years. So thank you all for having me. To follow his approach for clinical studies, the idea today was to try to talk about the preclinical world and what you have to do in order to satisfy the FDA for the requirements to get permission to test your product on a human. And so that's really what we're trying to cover today fairly quickly. It's fairly complex because we're not just talking about devices. So I was going to give a quick overview of all the funded projects with the Resource Center. Talk a little bit about which centers at the FDA have oversight over regenerative medicine products, and then how will the FDA regulate your product. A few preclinical development tips for devices, for drugs and therapeutics, for biologics, for cellular and tissue products, and oh yeah, let's hook them all together and call them combination products. What could be easier? And then a couple of slides on just making sure that you're enabling your submissions to the FDA to get permission to start your human trials under either an IND or an IDE submission. So I was very happy to find these graphics on the website for the Regenerative Medicine Resource Center. And it's interesting that there are 16 total ITP funded projects thus far. And look at the array of different types of tissue, different types of products that you are all developing. It is the most comprehensive and exciting set of 16 studies I have ever seen in my career. So how will the FDA regulate your product? I thought we'd take a really quick poll. How many of you know your product's regulation category? Okay, it's like eight of you. Very good.
Yeah, he doesn't know yet, so.
There you go. I believe you on that. How many of you know your product's intended use goal? That's very important, and if you don't know it, we're here to help you learn and define it. How many of you kind of know? I suspect there's many of us here that kind of know, but we're not certain. So, if you'd like to know for certain, how do we figure that out? We need to know the targeted patient population that you're aiming for. And how many of you have a preliminary regulatory assessment already conducted? There's a couple of you that are working on them. Excellent. This is one of the first things we really need to establish, along with, believe it or not, your marketing goals and your marketing plan and your intended use and your key clinical unmet need, so that you are developing the right product for the right unmet need. So how does the FDA regulate your product? The answer is really simple. It depends. If it's a device, these are some of the key regulations that you're going to have to deal with, and we are here to help you navigate through them, especially the quality system regulation under 21 CFR Part 820. That's also known as the Good Manufacturing Practices. The IDE, Investigational Device Exemption, that's how you get permission to start your human trial. And then once you get all the way through this continuum and you have finished your clinical study, we'll end up submitting all this data to the FDA under a marketing approval type submission, either a 510K or a pre-market notification. Or a pre-market approval. Okay. And actually, to be real honest, the device space is where I've spent most of my career, so I know it very well. And then, like some of you, we're all learning on this regenerative medicine continuum. Biologics and drugs and tissues are not necessarily as easy as devices. So if it's a drug, you'll have to deal with developing and manufacturing the drug per set of 210 and 211 in the IND, investigational drug application, and then eventually that would get approved through a new drug application. Is it a biologic? How many of you think you have a biologic? Anyone? There's one. I think there's more than one. There's two. Okay. It's a little harder to be sure that you have a biologic, I will tell you. So there are a couple of regulations for the biologics. Notice that the IND for the drug, 21 CFR Part 312, also applies to biologics. So biologics are in general treated very similarly to how a drug is treated from a regulatory standpoint. But the way you would know your IND is a biologics, it gets the two B's in front of it when you get the number from the FDA. Is it a human cellular product or a tissue product? How many think you might have human cellular? I know you do, Steve. Anyone else? There's one. Oh, Darnell, you do? Okay. Oh my, it's so much fun. So human cells, tissues, and cellular and tissue-based products. It's such a long designation that we call it HCTP. And this is probably, in my mind, one of the most challenging regulations and most challenging products to get through the FDA. And then, what if you have a device plus a drug, or a device plus a biologic, or a device plus human cells? These are combination products. And so they're also regulated through a separate set of regulations. And I'll just note that one of the first things we have to do if you have a combination product is to determine whether or not the primary mode of action is really device related, or if the primary mode of action is more like a drug or biologic. And so that's a real important distinction that we need to have clarified early on, because it will help define which center at the FDA will actually lead the review of your marketing approval. Okay. So, as I mentioned, we have 16 projects that have been funded. Wow, how many is this? How many? We got 9 devices. Now this is self-reporting according to Matsumi. These are the PIs that self-reported they thought they had a device. So we have 9 there. And then we have 1 cellular and 2 biologics self-reported. And then we have a total of 4 different combination projects. We probably may be updating this data as we learn more about the truth, whether you really have a biologic or a cellular. Okay. So this is just a very quick slide to just kind of make sure everybody understands at the FDA there are 3 main centers that oversee the regulation and oversight of medical products. The Center for the Center for Drug Evaluation and Research is CDER, if you have a drug-related product. CBER, Center for Biologics Evaluation and Research, handles biologics. And there's an important office to know about within that center, the Office of Cellular, Tissue, and Gene Therapies. Has anyone dealt with that office? There we go. We have a few people here. Okay. That office is probably one of the newer offices at the FDA. And so that you actually have people here that have actually interacted with them is a really good resource for everyone. And then, of course, the Center for Devices and Radiological Health oversee the devices and in vitro diagnostics, as well as radiological products. And then the under the CDRH is the Office of Combination Products. So as we've been talking about, this is a translational continuum, and the regulations speak to the continuum. The good laboratory practices are the very first thing that you'll be dealing with, and most of you are working towards your good laboratory practice checkbox, if you will. Once you've moved from the preclinical stage, then you're going to need to manufacture your product for your clinical study and eventually for the market. But how many of you are working with good tissue practices? GTP. I know Steve probably does. This is the least known of the GXP in my mind, but it's probably one of the more challenging sets of rules to handle tissues, especially tissues that have been harvested and now are going to be implanted into someone else. Very important to follow that if you have a tissue-related product. And then eventually these get moved into the GCP, the Good Clinical Trials Practices. Okay? And so not to overwhelm you, but this is pretty much everything you need to know. Put this under your pillow and you will be fine, okay? These are the different regulations. There are several regulations for clinical studies. Oops, oh, I'm so sorry. Sorry, hit the wrong button. Mostly we really want to talk about good laboratory practices in this talk. I'll be focusing on that. But just be aware that just about every type of product, whether it's a biologic or a drug or human cells, has a set of regulations in the Code of Federal Regulations. So how does the FDA know how to regulate your product? It has everything to do with you answering a series of questions about your product. How do you characterize your product? We have some projects that have a device-only project mode, and so it would be a device with a specific intended use using just a device. We need to know what the primary mode of action is, and what that means basically is the body metabolizing the product. Is it having a chemical reaction with the product, or is it more of a structural reaction to the body? And then what is the route of administration? Can you administer a biologic? Yes, you can. Can you administer tissue? Technically, you can. Can you administer a device? Technically, you can. So we need to understand where in the body your product's going to be utilized, and then what type of tissue is it touching, and for how long is it touching that tissue, and is it interacting with that tissue? And so this is a part of how we start building the characterization description of your product and how it works so that it performs as it's supposed to. We also want to make sure that we know, is it just touching mucosa? Is it touching bone? Is it actually interacting with blood as well? Because these questions help define the type of biocompatibility schema and testing you're going to need to do for your GLP preclinical data set. Okay? And something that often gets overlooked in the early stages is whether or not your product is going to be supplied sterile. Does it have to be sterile before you use it in the patient? Most of the products that we're talking about in this cohort will be sterile products. And if you think you don't need a sterile product, then we need to have a very strong rationale for why it does not need to be sterile. Okay? And if you forget about this, it means you also will probably forget about how your product is going to be packaged, and how it's packaged might be how are you going to deliver it to the clinical site of in the patient. For example, maybe it's a paste that's injected to the oral cavity. but you forgot that you needed to put it in a syringe, and you forgot that it needed to be sterile. So you want to be sure to address the packaging and sterility requirements for your product early on, especially before you do your pivotal preclinical animal studies, because the most important thing about the preclinical studies, if you don't take anything away from this 2 days, please take this away with you, and that is that the preclinical studies are required by the FDA so that we can demonstrate scientifically to the FDA that your product is reasonably safe in animals, and therefore it will reasonably be safe to try it in a human. Yes. Okay.
Some guidance on with the preclinical animal studies, whether you can get away with non-GMP agents that you test in the animals, and when you can and when you can't?
Yes, it's a very good question. It depends on where you are in your early research. So a lot of times in academic research labs, you're so early in the sandbox, if you will, that you just need to get some proof of concept that the idea works. From that standpoint, you don't need GMP, and you technically don't need GLP. But once you get to where you've evolved the product to the point where you believe you have a good candidate, and now this is the one we're going to run with, and we're going to take all the way down to the goal yard, the goal signpost, if you will, then you need to really pay attention to whether or not using a non-GMP manufactured product might have adverse effects on the patient. And so it's all about a safety risk mitigation approach that we take. And so if you have a very strong rationale for why you don't need a GMP product to be tested in your animal studies, then have that vetted, because there are sometimes a strong case to be made. But I will tell you, in most of the time I've been doing this for many years, it's typically the team that didn't follow GLP and didn't follow GMP approaches to making the products and testing the products that you will have to go back and touch it again and test it again. So it slows down your speed to market. Yes. Medical grade. Uh-huh.
But it's not medically grade, and the FDA will have you come back and retest it because medically grade is prepared under a CGMP facility, and it may actually in some ways change the property.
That's right. And you know, the thing that's so important about that, if you were to test it and you end up having problems in your first set of tests, it's very hard to trace back and understand the failure. Why did that product fail if you can't trace back to what went into that product? And that's really what good manufacturing is all about. It's about being able to trace exactly what that product is made of in detail. And I'll just go ahead and touch on the GLP question you asked earlier with Will's talk. It's a very important question, and there are some examples where a team can use, even in a large animal study, a non-GLP approach. I will caution you, though, if you are going to take that approach, you have to demonstrate to the FDA what about your GLP, your non-GLP study, what about that is not GLP, and why was it acceptable? It's the rationale for not using GLP is what you have to write up and demonstrate to the FDA. And there are some examples where the FDA will allow it. But I would tell you, please get some regulatory advice on that if you're planning to take that approach. There are some animal models that are just very hard to get into a GLP scenario, for example. But the other thing I want to stress, what does GLP really stand for? And I actually have a couple of slides on this. GLP stands, we say it stands for good laboratory practices, but I like to think of it as good scientific practices, because that's really all it is. We're documenting what we're doing, and we're protecting the safety of the animal. Is another reason GLP exists. Yes?
Just before we move on, so you mentioned 9 device companies or whatever. So you build a screw
Mhm.
and you do a preclinical study, and during that study you find out it's not as robust mechanically, and you'd like to enlarge the diameter.
Mhm.
Is that a reasonable adjustment, for example?
In a preclinical study, yes. In fact, that's a reason. That's one good reason for doing a preclinical study. And you're going to learn from the first few subjects of that study what's working and what's not working. And especially in the device development process, it's an iterative process. So it's acceptable to maybe stop. We learn something, go back and iterate the device, and then continue.
So it's a cautionary note. I've had CROs quote me biocompatibility testing. They say, "Oh, we don't need GLP for this," and so we've taken it to the Q-Sub meeting and said, "Okay, what are you guys willing to accept and get it, actually in writing from the FDA?" So they say, "Okay, for this particular leachable extractable test, you can do that non-GLP, but
Yes.
don't trust the CROs because they tell you everything is okay non-GLP, and because they quote you a cheaper price and they're trying to sell you something." So
That's right. The GLP, for a GLP protocol, you're going to pay a 25 to 50% surcharge, if you will, on each protocol because they have to follow quality requirements and demonstrate that they followed it. So, but it's a documentation package that will support your getting your clinical study approved as well. Yes.
So just maybe another quick perspective on the question. In my experience, at least, if it's a safety-related study that's going to be submitted to FDA, you're far safer. You're far more assured to do that GLP if it's
Absolutely.
reliant on safety.
Absolutely.
If it's a sort of an efficacy or mechanism of action preclinical study, probably don't have to do GLP. Another word on GMPs, I would strongly advise you to have a validated manufacturing process,
Yes.
meaning 3 validation lots after your engineering lot
Yes.
to take that product in to do your GLP tox and then, of course, your initial clinical
Absolutely.
trials, because
Absolutely.
otherwise you run the risk of FDA, in hindsight, looking back and say you didn't have your manufacturing process locked down.
That's exactly, we call that
And-
design freeze
Yeah,
and manufacturing freeze. Yes, that's very important. I'm going to move a little bit faster because I'm going to run out of time. I'm going to make sure you guys have a copy of these slides. So you'll have these definitions, but this is the definition of a device. And basically, it's, as I mentioned, it's primarily not achieving its primary purpose through a chemical action or being metabolized. Okay, so that's the key there to the definition of a device. Obviously, devices are regulated in a risk-based approach. The lowest risk classification of a device is a class 1. Class 2 is medium risk, and class 3 is the highest risk, usually life-supporting or life-sustaining type device. There might be a couple of class 3 projects in this group, and we want to know about that as soon as possible because it's a much harder road to hoe, if you will, for a class 3 device. And the FDA is going to scrutinize it much more carefully because it's a high-risk product. So what's a preliminary regulatory assessment? This is a tool that we use, and many different regulatory teams use something similar, and that is we're going to try to understand how does the FDA classify your product. In this case, this is for a device, class 1, 2, or 3. Is a 510K required? Is it a combination product? Which panel oversees of the FDA panels? And then what is the 7-digit code that actually describes your product? And what is the 3-letter code that describes your product? And then I would encourage you all to get a preliminary regulatory assessment, and the Resource Center is providing some support to your project. So that's something we've been helping some of the teams, and I think the sooner you have that, the sooner the Avenues company will probably be happy that you have that, because the marketing strategy and the regulatory strategy go hand in hand. You can't have one without the other. So the sooner you understand that and you have it kind of defined, it's going to help you. This is just a partial list of some of the key standards and guidances that will be helping you through several of these different types of products. The FDA has many guidance documents that are free, and they really speak to some of these very challenging regenerative medicine products and approaches. And that's especially what this list has, some of these guidances, and this is an active link. So a lot of reading, but a lot of it's free and available, and the FDA actually follows a lot of their own guidances. Usually, at least they try to, or they update them. And this is a slide you've already seen, again, trying to focus on the preclinical development aspects of preparing for the whole continuum to be successful. So I want to take a little moment and have the GLP talk with you, okay? Of all the GXPs and all the terminology that I see in academia especially, but even in industry, the GLP is the most misunderstood and feared GXP for some reason. And I've decided I'm on a mission to reduce the fear of GLP. GLP is really just good scientific practices. It's also a regulation that was put in place to protect the safety of animals. Do no harm to animals is also as applicable as do no harm to humans. And I come from the veterinary background, and I remember before that really was in place. And so I can tell you personally, doing no harm to animals is really important. So you want to minimize that. So that's what GLP really helps to achieve. And so you're going to get a free gift for attending this workshop. I'm going to give everyone their own GLP self-assessment audit questionnaire. This is what we use to assess a lab to figure out if it is GLP compliant. And it has everything in it you need. It has every single section of the 21 CFR Part 5.8, the GLP requirements. Notice we have general provisions, organization, and personnel. You need to qualify your GLP vendor. It's probably one of the most important few hours that you can spend with your GLP vendor. You need to document that you have qualified that they meet GLP requirements, and this tool can help you do that. I just took some key sections of it, but GLP covers your facilities, how you're handling your facilities, how you're dealing with your equipment. Are you calibrating your equipment? Your testing facilities, do you have standard operating procedures to conduct the studies at that lab? How are you controlling the test articles? That's very important. Are you storing it in a 22-degree freezer when it's supposed to be 0? GLP helps make sure that you're controlling your test articles correctly so that they don't get contaminated. And then your protocols for conducting your non-clinical laboratory studies, and then records and reports. So this is, I think it's about an 18-page survey, but you can utilize it, and at the end of it, you answer all these questions, and you can sign off on it. You can send this to any of the potential GLP partners that you want to work with and have them complete it and send it back to you. But probably most importantly, you can do this in your own academic labs. If you're GLP-like, and some of you have GLP-like labs in academia, which is great, you might be surprised. You're probably doing more of these things than you realize. And so get credit for it and document it. And then if you've got a few gaps, you'll know what the gaps are, and you'll have the opportunity to improve it over time, and pretty soon you'll be GLP compliant. So please don't be afraid of GLP. That's my message. Okay. So let's talk briefly about biocompatibility testing, which does take place in GLP laboratories. This is for medical devices in general, and this is how we figure out what type of biocompatibility testing needs to be conducted based on where the tissue, the material is touching the body, the surface of the device, what is it touching, the external communication of the device, whether it's a blood path, is it indirect? Is it touching tissue, bone, dentin? Notice dentin is in there. Who knew? And is it circulating blood? If it's an implant product, is it touching tissue, bone, or blood? And then based on the type of tissue it's touching, we talk about how long is it touching that tissue. Limited, less than 24 hours, prolonged, 24 to 30 days, or permanent, which is 30 days or longer. And so this is how GLP testing lab helps determine which cytotoxicity testing do you need to have, implantation test, hemocompatibility, and things like that. So this is another really important standard that's based on 10993, the ISO 10993 series. So many of you are probably working on this right now. How many of you are kind of working to figure this out? Okay, that's great. And you'll get quotes from these GLP biocompatibility test houses, and your quotes will range dramatically in price. And I probably stay really busy just helping my clients reduce the cost of their GLP biocompatibility testing. Sometimes you'll get a quote for $100,000 when I can get them down to about 20. So it's really, do you really need all those tests? Just do the test you really need. So there's some tricks to that. So I'll take a moment here to just walk you through. You remember Will showed you how the continuum of clinical trials works. This is how we get to where we have permission to start a clinical trial. For a device, we design and develop the device per the regulations. Then we freeze the prototype design, and we conduct a design verification to make sure we designed the product correctly. And then we move it into bench testing. So this is in vitro testing to show that the product is performing reasonably well in an in vitro bench engineering environment. Once you pass that, then it's time to get your GLP vendor qualified and work on your animal protocols. So I want to take just a moment to make sure that you understand in designing your preclinical animal protocol, you also want to keep in mind the long-term goal, which is supporting the primary endpoints of your clinical study. The animal protocol should line up with your goals for your clinical study. And it's probably the one thing that many teams kind of forget to do. You only want to do these animal studies once, or if at all. Some of these products you may not need animal studies, but most of you will. And so if you're making sure you're keeping up with the biostatistician in your preclinical protocol design to feed into your human protocol design, you will save a lot of time and money and not having to go backwards, okay? So once you gain permission to do the study, for the animal study, you run it, conduct it, then you summarize it, and you find out, did we indeed get the answers that we wanted? Did it perform as intended? And did we meet our primary and secondary endpoint goals of the animal study? Once you get a yes to that, then you call Jeannie Wright at my app, and she will help you, and her team will help you prepare your IND or your IDE submission to go to the FDA, at least here in Ann Arbor. And this is just a graphic to try to give you a headache. When we talk about submissions, there's a lot of submissions to get to the end goal. If you think about it, some of you will be having pre-IDE meetings or pre-IND meetings. You might have a first one, and you're allowed two. So you might do that. You have to actually provide a whole packet to the FDA. Some of you, I think, have already done this successfully. And then once you go to the FDA to talk about your proposed clinical study, let's say you get it cleared through the FDA, and now you have to submit it to the IRB and make sure your institution is approving that study. And then eventually you move into your human pilot study for a device, and eventually your pivotal study, okay? But for the purposes of this talk, this is where we are. So we've still got a lot of work ahead of us. That's sort of what the point of this slide is. But you will not succeed in any of these submissions if this is not done correctly. And so it may take longer than you want, but if you can focus on this early preclinical design of your studies and all the requirements, it will really serve you well. All right, so, and that's just one submission to get permission to start one clinical trial. And Jeannie and I worked on this one together. So it was about 1,200 pages. So the FDA reviews an awful lot before they allow you to do a human study. Okay. All right. I'm going to run through the rest of these slides, and you will be getting a copy of them. But the development process is pretty straightforward with devices and drugs. With biologics and so forth, you're going to have your chemistry, stability, and your analytical development done, your non-clinical toxicology and PK and pharmacology, and then getting your submissions ready. So this is just a quick checklist to remind you to make sure you have a clear intended use statement. When you have that, that defines your regulatory pathway requirements. Make sure you get a preliminary regulatory assessment, and then you qualify your GLP vendor early. Do early preclinical planning. Get some support on that. Preclinical testing to support your good clinical practice human trial. What is your delivery route? How long is it going to stay in the body? And if it's biodegradable, you need to understand that and characterize that very, very scientifically for the FDA. Let's see, we talked about most of these things. GLP compliant or GLP light or like. Now you have a tool. After I give it to you, you'll have a tool to be sure that you're doing GLP. And if you're not going to follow GLP, you need to have a gap rationale for what part of your study did not comply with GLP. And then make sure you plan for your sterility, packaging, shelf life testing. Okay. Same idea, you've already seen this slide. These are the keys. In order to get all the way through this continuum, the green box is probably the most important part of your journey. What is a biologic? Okay, now this is a very long definition. I'm not going to read it to you, but I have it in here, okay? There are guidance documents, that's the key here. Non-clinical studies are very similar to drug studies, but there's some additional things, as Steve mentioned earlier, that you have to test for. And it's usually on a need-by-need basis. Biologics are very unique, and the FDA doesn't have it figured out. And so we have to work with the FDA to get the best answers. And that's why early meetings with the FDA around a biologic product is a very good idea. And it can keep going here. So the main difference between biologics and drugs has to do with the size of the molecule. Biologics are typically much larger molecules. They have different dosing requirements. They're very species specific, and so what kind of species, what kind of test model you're going to use becomes very important. Again, where is the overlap between a drug and a biologic? This kind of demonstrates real quickly where there's some similarities, but there's some uniqueness to the biological continuum. And again, it's a long journey. This is just some examples of the types of things you have to deal with in a biologic translational development program. So CMC, some of you know what that stands for. It's chemistry, manufacturing, and controls. It's used in the drug terminology and also in some of the biologics. These are some of the key things you have to do to make sure that you're developing your current good manufacturing practice for your biologic product. And then, of course, that will support your preclinical GLP studies, and then that gets submitted to the FDA for permission to start your clinical trial. I mentioned this earlier. With drugs, it's usually 2 species are required. With biologics, it's often 1 species, and unfortunately, there's often just 1 species that the FDA will allow, and it is usually human primate, the primate, non-human primate model. And we have a very seasoned investigator here who's very familiar with working with the non-human primate model. Again, same idea. This one, All right. What is a human cellular and tissue product? It consists of human cells or tissues. How many of you think your product will contain human cells or tissues? You're our biggest challenge, Steve. You take on the hardest projects, but could be really exciting. And yours, maybe? Okay, maybe. I noticed you had human and now you're moving to bovine. That may be a really good idea. But we might want to look at that a little bit further to make sure. There are many things that are not HTCPs, and that's part of the definition as well. So again, there are many guidance documents. I encourage you to download these and get to know them. They have a lot of good guidance in how to handle your type of product. And a lot of them have, decision trees that you can walk through. Preclinical program objectives. This is taken directly from that guidance I just showed. I won't spend time on it, but I just want you to be aware of this. This information is available, and if you're not using it, please start using these guidance documents. And then moving into combination products real quick. I probably have 60 seconds maybe. Keeping in mind that a combination product is going to be a challenging regulatory path in that it could be a device and a biologic combination. It could be a drug and biologic. It could be a device, drug, and biologic all combined. Let's hope not, but it could be. And so you're going to need extra regulatory support, I believe, to be successful with a combination product. And I hone in a little bit here on what does an IDE contain. This is just a table of contents of what the FDA requires for an investigational device exemption. So that you're familiar, this is the big submission we'll have to prepare for you to get permission to start your clinical study. And I'm really going to hone in on this second section, which is really known as ROPI, the Reports of Prior Investigations. As you'll see, it's talking all about your preclinical efforts, and look at how much the FDA wants to see about your preclinical work before they allow you to start that in a human. Yes.
Thank you very much. In your preclinical, thank you. In the preclinical animal studies, when you're analyzing your data, do you need to keep in mind ways to be blinded in interpreting your results?
Absolutely.
And how?
Yes,
do you
and that's
do
part of
that?
the GLP practice.
Oh, okay.
Okay. We can talk further, but that's a really good question. And yes, because you don't want to be biased in your decision making, right? Yeah. So this is, I think, what the last slide, and the main point of this slide is, what does the FDA review when they look at your IND or your IDE submission? They're going to review all of this information to decide, is it okay, is this product reasonably safe to allow this clinical team to study this product in humans. And I think I have, yeah. And so your preclinical work is a big section of what the FDA reviews. I would say they spend as much time on the preclinical work in this FDA review of your proposed clinical study as they do with your protocol for your clinical study. And the reason for that is if they don't catch it, and you didn't catch it in the animal study, and they allow you to go forward with this human study, they might miss something, and it could lead to serious adverse events and harm in patients. And so that's why they spend a lot of extra scientific review time. They have a whole team that reviews your preclinical work during your clinical trial review process. And so, their mission is to protect the public health. And I found over the years, I think many of you have worked with the FDA. It's a really incredible team of scientists and clinicians and people that really do have the public's interest at heart. And they also want to work with you. In particular, the NIH funding this particular program, I think, is a really wonderful opportunity to get regenerative medicine into the field in small examples, if you will. And if we can be successful with these technologies, then we'll open up the same technologies for different parts of the body. And I'm so excited to be a part of this team. And any way I can help you, please let me know. That's it. Okay. Yes, you have a question?
How often does the FDA disagree with the investigators' claim on the primary mode of action for a combination product?
Boy, that is a fabulous question. I don't know how often, but I will tell you, I didn't really talk about this, but the way that you have that discussion with the FDA is you can submit a request for designation. And it all has to do with that. What goes into a request for designation has very specific questions. And in those questions, you are describing your product as you understand it. So you have the science behind it to explain what you believe is the primary mode of action. And when you present that to the FDA, then they review it and they may push back on it. Okay. And so part of it has to do with they may not agree with it if it's not well characterized, or they believe there's really some scientific errors in what you're claiming is your primary mode. And a common example of that is if you think of the drug-eluting stent for cardiovascular purposes. What's the primary mode of action? Is it really a mechanical device and a stent to open the vessel, or is it the drug-eluting element of that product? And early on, when those products were first coming out, there was a lot of back and forth for that example, where convincing the FDA it was more of a device primary mode rather than a drug primary mode. So I would say the best way to get the FDA to agree with you is to have solid science to demonstrate that your primary mode is the way you say it is.
That was a great talk. Thank you. You talk about design freeze before benchtop testing or preclinical testing. Is it necessary to have kind of blanket design freeze, or is it possible to have design freeze with respect to elements that are going to be tested in a given test? So if you have material design freeze for biocompatibility.
Yes, in fact, to call it design freeze for your animal study is probably a little I probably misspoke a little bit. We like to call it design slush, like a Slurpee. So you're close to being done, but you recognize you're going to learn from that study that's going forth, and you may have to come back and polish and iterate the design a little bit, whether it's the material or the performance mechanisms. So
if you change the design, for instance, after your biocompatibility testing, but you don't change any of the materials, would you expect to have to go back and redo biocompatibility testing? Or the same thing for software or
great question. So anything, anytime you change the design, let's say you finish your animal studies and you had to modify the product a little bit, you do a regulatory assessment to see if that change triggers the need to go back and restudy it. So did it change the potential for safety and efficacy? Is there another potential risk that you hadn't identified? And if you can answer all the questions that we haven't really changed the form, fit, or function, then you likely won't have to change it. Does your product use software?
Yes, it does. Yeah.
Oh, talk to me. I didn't even mention software, but let's talk.
Okay, great. Thank you.
Any others?
Yeah, Kay,
Yes.
thank you
Hi,
for
Steve.
the comprehensive overview. And
You're welcome.
I just wanted to try and help clarify something. So you asked a key question in our session this morning, whether our device was a device of non-significant risk. And I was trying to understand how much of the more complicated side of what you talked about relates to more the significant risk devices. And then secondly, I wanted to just share that when we first were in the process of licensing the technology from the Office of Technology Transfer, we dealt with some reviewers there that sort of pushed us to do animal studies, because in medicine you do animal studies, but in dentistry we were actually led to believe by some investigators that there really aren't that many valid animal models for caries, dental caries evaluation.
Good point.
So, and you also told us that anytime you can do it in vitro, it's better. So when
That's
we
right.
visited with the FDA, they agreed that in vitro was acceptable.
Yes, I would say for many dental products that may be the case, especially your goals. As far as the FDA's real approach is, if you can study it and prove its performance, that it's performing as intended in the bench, in an in vitro scenario, or with phantoms and modeling, they prefer that. We want to try to reduce how many animals we're having to study, and eventually, actually, FDA doesn't even want us to have to test these things on humans if it's not necessary. So having a really robust benchtop phantom model mechanism in the dental blocks that you were talking about, I think is very compelling. As far as this significant risk versus non-significant risk study, I'll just touch on that briefly. It's a determination you have to make to tell the FDA whether or not, or the IRB, whether or not your product has to both have FDA review as well as IRB review to get permission to start the clinical trial. I was focused on preclinical. Yeah. But we have a tool, and Jeannie's team also has a tool. It's just a questionnaire that you answer to help you triage whether or not it's a significant risk or non-significant risk study. The key is the significant risk study has to have both FDA and IRB approval before you can start that clinical trial.
So are you better than to seek the clearance before you do your clinical trials?
I would say just it would take someone less than an hour or two to fill out this this questionnaire to know whether you're going to be a significant risk or not. Mm-hmm. And even if you're a non-significant risk study, you have to submit that to the IRB to get permission to start your non-significant risk study, and they have to agree with you. If they don't agree it's non-significant risk, then by default you have to go to the FDA and get their opinion. Yes?
Another one? Oh, here it is.
There you go.
Thank you.
A voice from afar.
Yeah, I really appreciate the fact that you want us not to be afraid of GLP, but you know, our institution does not allow us to brand the label GLP. They just don't allow us. So let's assume we function in following the GLP guidelines in our laboratory. You establish all the processes, but as soon as you approach somebody who's leading a project and you say, well, we can't be GLP, right away they're a little bit afraid, and they say, well, the FDA wants GLP, and then, you know, that's a big risk for our project. What is this should be brought up in the pre-sub with the FDA, or what is the risk of going down this path?
What is the risk of going without GLP?
Correct, but following GLP guidelines, but you don't have the official label.
I would say that the way I would answer that has everything. It depends on what kind of product you have and some very specific details to know whether it would make sense or not. And a Q-sub type meeting that you're talking about, having a meeting with the FDA, FDA can give you their opinion in those meetings, but everyone needs to be aware that those are not legally binding opinions. They're only legally binding when you have submitted formally to the FDA either your IDE application or your 510K application. And so the team that tells you that when you're meeting with the FDA early might not be the same team when they're reviewing everything. So take that with a grain of salt.
So then we shouldn't.
GLP is required by law. I will just clarify that for everyone.
So then it is a slippery slope.
It's in the law
that
you're supposed to follow GLP. If that doesn't, you know, so that's when we get to the regulatory science FDA hat, you know, they have to make sure you're complying with the law. And if you're not going to comply with GLP, then you need to identify what parts you did not comply with and why it was okay, and give them that rationale.
Okay, thank you.
Question, can you even get the certificate for a GLP? And if you can't, then is it even worth it to do these experiments in-house, if it's required by law to have the GLP certificate for your data? So would it be better to just find a vendor and get your data generated by that vendor and period? So, you know, how how much of that judgment has to go in deciding what needs to be done? For instance, how do we spend the $100,000 that we get now,
Yes.
as opposed to just, okay, that's what we need to do, but it's useless, so let's get somebody else to do it with the GLP certificate.
So another way to answer this pretty quickly is in the early research, if we think of research and development, if you're in the early research in your academic lab and you're just trying to get proof of concept, that you do not need to be GLP if you're doing a small animal study, for example, to show that it works reasonably in your proof of concept. It's when you move out of research and you're moving into really developing and polishing the product, that those studies that would be supporting the animal proof of safety should be run under GLP, because that's going to support the argument you're making to the FDA that it's going to be reasonably safe to test in humans.
So it needs to be repeated one way or another under GLP.
Usually the small animal study to proof of concept is not the same as your GLP study, which might be in a larger animal model and would be probably more animals. Okay, so that's really your seminal data to prove to the FDA it's reasonably safe, and that's when you really ought to try to do GLP. Okay.
Quick point here in the back.
Yes.
Thanks, Kate. That was excellent, and I want to
you.
just underscore something both you and Will said about the importance of integrating this regulatory development strategy with marketing and clinical assessments, and honestly, product preclinical models even come into that mix, and business development. It all has to be really connected from the beginning.
Absolutely.
And I think sometimes, especially in academic programs where there's a lot of respect for the expert and the expert's siloed away somewhere and they go speak with that expert and then go to a different expert in a different silo, and it's left to a person who may not have had a lot of experience commercializing technologies to connect all those dots. And the success or failures happen when the dots get connected.
Yes,
that is
you're
what
absolutely right.
makes
I love
these
that.
things commercially viable. So I just really want to stress that for those of you who aren't working in a way where you're putting people together around strategy and thinking about all of these things early on, because once you're, even with your preclinical model, you may be off to the races in the wrong direction.
That's right.
So,
that's exactly right. I applaud that. And actually, sometimes in industry, marketing and regulatory, people say they have their oil and water, but actually marketing and regulatory work hand in hand. And you need a cross-functional team of experts, manufacturing, regulatory, clinical, marketing, to be able to develop a product that is going to be not only performing as intended, but meeting that unmet need, and it's going to be highly reliable. Remember, you want people to buy it again.
Right, and your goal is a commercially successful product. I mean, I think sometimes if you're focused too much on the FDA, especially with these really onerous long-term approval processes, if you're focused too much on getting that as if that's sufficient, it's not. It's necessary, but it is not sufficient for commercial success. So, you know, the FDA piece of it has to be really a piece towards a longer-term goal of commercialization.
I applaud that. You're absolutely right. Remember, it's one thing to get it approved, but if there isn't a big enough market or there's no reimbursement strategy for how you're going to get reimbursed, we're not even talking about reimbursement for some of these because it's dental. Dental isn't as reimbursable, but some of these regenerative therapies might be reimbursable, and we might want to have some workshops on how to get reimbursement codes through the CMS, which is a whole another journey, frankly, but it's a very good point. Thank you. Are you marketing? Yes. Oh, Jeannie, yes. Okay. God, I see you. I see you. She followed me at Gore. I'm sorry. I wasn't sure that was you.
I think we're all ready for a break, but I just want to throw out for everybody's benefit the shift in biocompatibility with the L&E kind of being a new buzz for FDA, and they really want a lot of leachable and extractable studies. I mean, it's not in your standard chart, but they seem to be asking for it. I don't know what your...
It depends on what your product is. What is your product?
Well, it's bioresorbable.
Well, that's why. Many products aren't resorbable, so you're right. And if it's a bioresorbable product, then you need to understand how long does it take for it to be totally reabsorbed, or is it totally reabsorbed, and what does that profile look like? Because that's all about safety. But you're right. Yeah, that's a challenging. That's a new material typically. Thank you. One more?
One more question. Sorry. I'm involved with a clinical trial that involves a drug that has been in phase 3 clinical trials, and we have access to the GLP tox, and all of that information has been allowed to be used in our filings.
That's congratulations. That's a significant achievement in itself.
Right. So, however, we're not delivering the drug systemically. We're delivering it locally in the mouth.
Okay.
And so my question is, so normally what would be done, I would say, if you didn't have all of that at this stage, is you would have your GMP formulation, and you would do a GLP tox study for that. So
because your route of administration is different.
Well, I'm just saying, irrespective of whether we have that phase 3 clinical data or not, we would have to do a GLP tox. So the question is, now that we have that data, do we still have to do the GLP tox for the local toxicity?
I will have a sidebar with you to maybe ask a few more questions, but in general, if the route of administration is different than all that other data,
right,
and it sounds like it is,
yes, it is,
then it's likely you'll have to do at least a small study,
a small GLP tox, particularly on the formulation.
Because if the route of
toxicity.
administration for the phase 3 trial is injectable, for example, it's systemic.
Yes,
that's more dangerous.
Yes,
if it's a route orally, that's limited, and the dosing is much smaller, probably all those things, it's probably going to be safe, but you still need to demonstrate it. And I would think it would be a small subset of data that you would need under GLP for that.
That was my impression. Thank you.
Good idea. Thank you. Thank you all.